morning students. So today we are going to continue with the enzyme portion. Okay, so the first we have already discussed about enzymes to some extent. Thereafter we learned about what is activation energy, and then we saw regarding the factors which affect the enzyme action. In that we have already covered three factors, right? So one is the temperature. Optimum temperature is the best where the enzyme is actively participating. Below that as well as above that, when you are seeing the temperature below the optimum temperature and above it, or below the optimum pH or above it, the enzyme activity is less, isn't it? So this is regarding temperature and pH, which are the factors which were affecting. And then we saw about the concentration of the substrate, isn't it? So in that also we saw when the concentration of the substrate goes on increasing, the rate of reaction also goes on increasing, but after some time when you see it becomes constant. Constant means there is no more enzymes to get attached to the substrate to speed up the reaction. So the rate of reaction remains constant. So there I can increase the rate of the reaction only when I add what? Some more enzymes into it. Otherwise the rate of reaction will be remaining constant. Okay. So till that we have already discussed in the next last class. So now we are moving on to the next factor. So what is the next factor when you are seeing? It is the inhibitors. So this can also be considered as a factor. Inhibitors, presence of some of the inhibitors which disturbs the enzyme action. Okay, so the enzyme, in order to act, some inhibitors are there which can disturb the rate of the chemical or the metabolic reaction. Okay, so what are these inhibitors? How are the enzyme getting inactive, or how is the enzyme not functioning? Listen carefully. The inhibitors are something which is making the enzyme deactive, so that the enzyme is not acting as a catalyst anymore. So this inhibitors, when you are seeing, can be of two types. So what are the two types of inhibitors? So inhibitors क्या है? जो enzyme को उसका काम करने के लिए inhibit करता है, करने नहीं देता, ठीक है? तो that one we call it as what? We call them as inhibitors. These inhibitors, how many types are there? There are two types. What are the two types? One is your competitive inhibitor and the second one is your non-competitive inhibitor. Okay. So what is the meaning of competitive inhibitors? So these are some chemicals which you are seeing, they are resembling somewhat to that of the substrate. So this is the substrate which goes and binds with the active site of the enzyme. Imagine that is one substrate which is actually the substrate which wants to go and bind with the enzyme there in the active site. But there is another one substance. This substance is having the structure similar to that of the substrate. Now what will happen? Instead of this substrate, this substance which is similar to the substrate is going and attaching with the active site of the enzymes. Now what will happen? The substrate cannot get attached because another substance has got attached to that active site. Now the substrate is not attaching, so the reaction will take place? No, it will not take place. So competitive inhibitors are one which comes into competition, inhibiting the enzyme action. Why? Because this competitive inhibitor, when you are seeing, is having the structure similar to that of the substrate. So when they go and bind to the active site, this substrate cannot go and bind. So the enzyme action will not take place. Okay. So these are chemicals that resembles an enzyme normal substrate and compete for the active site. Okay. Excuse me. So that is your competitive inhibitors. Now next one is your non-competitive inhibitors. How do they hinder the enzyme? वो एंजाइम को क्यों काम नहीं करने देता, ठीक है? नॉन कॉम्पिटिटिव इनहिबिटर्स, व्हेन यू सी, दे डू नॉट गो एंड अटैच टू द एक्टिव साइट. They are not going to go and get attached to the active site. So where are they going to get attached? When you are seeing on the enzyme, on some other place they will be going and getting attached. So after getting attached, this non-competitive inhibitor, it is a chemical substance which when goes and attached with any of the site of the enzyme, what this enzyme bonding breaking takes place, and the structure of the active site, what happens, get altered. Altered means it changes. So when the substrate is like lock and key, okay, if the key as well as the lock fits in only when it is of particular structure. If the structure is changed, then what will happen? They cannot fit in, isn't it? So non-competitive inhibitors is like that only. They are going and attaching with the enzyme, thereby changing the active site, structure of the active site is changed. So when the active site is getting altered, it's getting changed. Usually how it was like this, isn't it? Like this it was. Now it has altered and it has got a structure like this. Now what will not happen? The substrate cannot go and attach and the enzyme action will not take place. Okay. So this set of inhibitors we call it as what? Non-competitive inhibitors. Active site पे जाके नहीं attach होता. ये non-competitive inhibitors जो है वो एंजाइम के किसी दूसरे जगह पे जाके क्या हो जाता है? They will go and get attached. अटैच होने के बाद क्या कर देता है? एंजाइम का स्ट्रक्चर बदल जाता है, एक्टिव साइट का स्ट्रक्चर बदल जाता है, तो सबस्ट्रेट जाके वहाँ पे लॉक की जैसा अटैच नहीं हो पाता, तो ऑटोमेटिकली एंजाइम का काम नहीं हो पाता. ओके, सो दिस सेट ऑफ सब्सटेंसेस वी कॉल इट एज व्हाट? नॉन कॉम्पिटिटिव inhibitors. Okay, so two examples of enzyme inhibitors are there. One is competition, so they are having a structure similar to the substrate, so they go and attach to the active site. Non competitive, they do not have a structure similar, but these substances goes and attach to any one of the enzyme place. Okay, thereafter they alter the active site so that the process is not taking place. Understood? The next topic what we are going to discuss is the chemical composition of the enzymes. Okay, so when you are seeing the chemical composition of the enzymes based on the what all compounds it is present, the enzymes can be of two types. So what are the two types of enzyme? It can be of a simple enzyme or it can be a conjugated enzyme. So what is the difference between the simple enzyme and conjugated enzyme? Okay, simple enzymes, when you are seeing, the whole enzyme itself is a protein. There is nothing else other than protein. The whole structure is composed only of protein. Okay, so if it is totally protein itself, then we call them as what? The simple enzyme. Like you have the enzyme like pepsin which digests protein which is present in the stomach, trypsin from the small intestine which is digesting protein. So these are all examples of enzymes which are only proteinaceous. They are made up of only proteins. Okay, another one enzyme is your conjugated enzyme. This conjugated enzymes, when you see, what is the difference? They are having two parts. So what are the two parts of the conjugated enzyme? One is the protein part and another one is your non-protein part. Okay, so the protein part of the enzyme we call it as what? Apoenzyme. So conjugated enzyme is having how many parts? Two parts. So this protein is one part and another one is non-protein part. Only with this protein part will not functional. The enzyme will only become functional if it is having this non-protein part as well in conjugated enzyme. So वो functional कब होता है? जब non-protein part भी रहे तब भी ये protein क्या होगा? enzyme के जैसे काम करेगा. So this non-protein as well as protein part of the conjugated enzymes together we call it as what? Holoenzyme. What do we call them? Holoenzyme. Then only the enzyme is getting functional. Okay, now this protein part of the enzyme is called as what? The apoenzyme. So what is the protein part of the enzyme of conjugated protein? It is apoenzyme. And what is the non-protein part called? The non-protein part is called as cofactor. What do we call them? Cofactor. Okay, this cofactor when you see is of two types. What are the two types of cofactors? One can be organic, which is made up of carbon, hydrogen and oxygen more composition. So that is organic. Okay, and another one is your inorganic metallic ion. So one is the organic part cofactor and another one is your inorganic part. This organic part, when you see that non-protein part which is organic in nature, is of two types. Okay, so what are the two types of these organic non-protein part when you are seeing? The organic cofactor when you are seeing, it is of two types. So what are they? One is one that can be easily separated. The enzyme can be easily separated from that cofactor. जल्दी से क्या हो जाता है, वो दोनों को सेपरेट कर सकते हो. प्रोटीन एज वेल एज द नॉन प्रोटीन पार्ट को कैन इजीली सेपरेटेड आउट. ओके, अनदर वन इज कांट बी इजीली सेपरेटेड. और प्रोटीन और जो कोफैक्टर रहेगा वो दे आर वेरी टाइटली बाउंड and it is very difficult to separate them. Okay, so one which is can be easily separated from that of the protein and the non-protein part separately, I can take it out. These enzymes when you see, we call it as coenzymes. What do we call them? We call them as coenzyme. For example, NADP. NADP, the full form when you are seeing NADP, you will learn in photosynthesis, respiration and all this enzyme. Nicotinamide adenine dinucleotide phosphate. Okay, so this nicotinamide adenine dinucleotide phosphate is containing a the protein non-protein part called as one of the vitamin niacin. Niacin is present along with the protein part. Okay, so that is an example for a coenzyme, and it can be easily separated. The protein and non-protein part, you can easily separate it out. Okay. Another one is your can't be separated. Protein part and non-protein part are tightly bound. Okay. So for that, the example is what? Phosphatic group. So I can divide the conjugated protein into two. Who are they? Protein and non-protein part. Protein part को क्या बोलते हैं? Apoenzyme. Non-protein part we call it as cofactor. The cofactor is of two types. One is organic and another one is inorganic. This organic, when you are seeing, it can be of two types. One which can easily be separated and one which cannot be separated. Separated क्या मतलब? वो protein और non-protein part को easily separate कर सकते हो. Cannot be separated, you cannot separate the protein and the non-separate part easily. Okay. So example for the separatable part means which you can easily separate it out, that one we call it as coenzymes. Which cannot be separated, that is phosphatic group. Phosphatic group example is your peroxidase. So this peroxidase, when you are seeing, it is containing a non-protein part, heme part. Okay, and this heme phosphatic group is helping in the reaction, like hydrogen peroxide is converted into water and oxygen. It's broken down into water and oxygen by this enzyme peroxidase. It contains a heme part. Okay, so that heme part is the non-protein part of the protein enzyme. Okay, so this phosphatic group is another one. So what and all we have learned? Cofactor, दो तरफ का cofactor पढ़ लिया है हम लोग. कौन कौन सा है? One is your coenzyme, second one is your phosphatic group. The third cofactor is your inorganic metal ion. Inorganic one, under that activators. What do we call them? We call them as activators. Okay, like for example, an enzyme called as carboxypeptidase. Okay, so this carboxypeptidase, when you are seeing, it's an enzyme. In that, one of the inorganic metallic ion is present. What is that? Zinc is present there. Okay, so the zinc, when you are seeing, it is a metal ion which is present in the enzyme, and they are coming under the category of inorganic metal ions, and these are called as what? Activators. So hope it is clear. So cofactors, name the cofactors if they are asking. Three cofactors you are learning. What are they? One is your coenzyme, second one is your phosphatic group, and third one is your activators. Okay, so or inorganic metallic ions, that also you can say. Okay, so this cofactor, if you are seeing, can be divided into two. What are they? Organic, inorganic. Organic में दो आता है. One is coenzyme, second one is phosphatic group. Inorganic, what is it? Metallic ions. Okay, so otherwise they are referred to as what? Activators. Okay, so these all examples you have to just learn, and you should understand what it is. Okay, so this is the chemical composition of what? The enzyme. Next we are moving on to the classes of enzymes, means classification of enzymes. Okay, excuse me. So we are going to classify the enzymes. There are different enzymes, like mostly when you are seeing, when you are writing you are having enzymes like lipase enzyme, isn't it? Isomerase, like that many enzymes are there. All those things we are not going to discuss. We are going to discuss only based on biochemistry. Okay, International Union of Biochemistry has classified enzymes into 6 categories. How many categories? 6 categories. Only those classification we are going to deal with. Okay, so the first category of those enzymes under biochemistry, 6 classes are there, means 6 groups are there. So we are going to learn all these 6 groups. Okay, the first group of enzymes, first classification under that, the first group of enzymes is all these based on their functions. What functions they are carried out, based on that they are named. Okay, oxidoreductase. What is the first enzyme? Oxidoreductase. First types of enzymes are oxidoreductase. So what is this oxidoreductase? As the name suggests, they are undergoing oxidation and reduction simultaneously. So you know what is oxidation, addition of oxygen, removal of hydrogen is reduction, that and all you know, isn't it? So electrons, flow of electrons, or electrons is either accepted or is being given out, so that one we call it as what? Oxidation and reduction reaction. So when one substance is getting oxidized, automatically another substance will be getting reduced, isn't it? So that sort of reaction which is helping in both oxidation and reduction, those enzymes which are playing a major role in that sort of reactions, we call them as what? Oxidoreductase. Okay, like for example, you see PGAL, that is phosphoglyceraldehyde. Okay, so this is in respiration you will be learning in detail. So phosphoglyceraldehyde, when you are seeing, it is combining with nicotinamide adenine dinucleotide. So here what is happening, this is an aldehyde. PGAL is what? Phosphoglyceraldehyde. So aldehyde, you know what is it? CHO, isn't it? It is getting converted into 1,3-DPGA. What is DPGA? Diphosphoglyceric acid. So this diphosphoglyceric acid is what? COOH, isn't it? So CHO to COOH it is getting converted. Here it is CHO aldehyde is getting converted into acid, COOH. So what is added? Oxygen is added. So what is the reaction? Oxidation. So this one is getting oxidized to this. But here you see NAD is present. It is getting converted into what? NADH2. Means here what is actually happening? NAD which is getting converted into NADH2 means hydrogen is added, isn't it? So here what sort of reaction is taking place? It is reduction. Okay. So one is oxidation, another one is reduction. So you can find a compound is getting oxidized and another compound is getting reduced. So this sort of reaction we call it as what? Oxidoreductase. Okay. So these enzymes are coming under oxidoreductase. What is the enzyme which is involved in this reaction? Phosphoglyceraldehyde dehydrogenase. So this enzyme is helping in the conversion of phosphoglyceraldehyde to diphosphoglyceric acid. Okay. And NAD is converted into NADH2. So there what is the reaction taking place? Oxidoreduction reaction, oxidation as well as reduction, and the enzyme involved is phosphoglyceraldehyde dehydrogenase. What is the type of enzyme? That one? Oxidoreductase. Okay. The next is your transferases. So second category of enzyme is what? Second one is your transferases. So as the name suggests, transferases transfer, they are helping in transferring the groups, okay, transferring any one of the group from one to another. Like for example, if amino group is transferred, then it is aminase. If carboxy group is transferred, means the carbon dioxide, then it is carboxylase. So like that, transfer of group is taking place. So that sort of enzymes which is helping in transfer of group when the reaction is taking place, those enzymes we call it as what? Transferases. जो transfer करने में help कर रहा है, किसको transfer करने में? Group को transfer. Like here example it has been given. So what is the example given? Glutamic acid is reacting with pyruvate. Okay. So it is again, you can find all these reactions in respiration, photosynthesis and all you will be learning this. So just for understanding I am giving you. Okay. So glutamic, you need not learn the examples and all, you should know what it is. So transferases, when you are seeing glutamic acid is reacting with pyruvic acid, it is getting converted into. So here amino group from the glutamic acid, कौन सा group जा रहा है? Amino group from the glutamic acid is transferred to the pyruvate group. Okay. So when this transfer of amino group NH2, isn't it? Amino you have learned, isn't it? So amino acid when you are learning you will have learned. So this amino group from glutamic acid is transferred to the pyruvate. Okay. So what will happen? The reaction will be taking place and you will form what? Alanine and alpha ketoglutarate acid or alpha glutarate. Okay, ketoglutarate will be formed. So this here what is happening? Transfer of amino group from glutamic acid to pyruvate. That's why the name of the enzyme is glutamate pyruvate transaminase. Okay. So ये glutamic acid glutamate same है, pyruvic acid pyruvate same है. Okay, that is same. You can either use it as pyruvate or you can say it as pyruvic acid. You can say it glutamate or glutaric acid. Okay, that both are one and the same. So here from here the amino group is transferred. That's why the enzyme which is involved in transferring this amino group from here to here and resulting in the formation of this product, those enzyme we call it as what? Glutamate pyruvate transaminase, since the amino group is transferred. Okay, so this sort of reaction we call it as what? Transferases. Sorry, the sort of enzymes we call it as what? Transferases. Okay. The next enzyme is your hydrolysis. Hope it is clear. Oxidoreduction में क्या होता है? Oxidation reduction. Transferases कौन से enzyme में react होता है? Where the transfer of group is taking place. And hydrolysis, where it is reacting, means where you can find this enzymes are involved. Hydrolysis, where there is a breaking of bond by water molecule. Okay, water is added and this water is helping in breaking the bond and forming compounds. Then I call them as what? Hydrolysis. Addition of water is helping in breaking of the bond, resulting in the formation of some substance like starch is digested by amylase. So for digestion and all, mostly you can find this one, and it is converted into maltose, isomaltose, limit dextrin, like that. Small monosaccharides it will be converted into, isn't it? Monosaccharide, disaccharide, like that. The starch is broken down. So what is the enzyme? You know, either it is in the saliva, salivary amylase, or in the pancreas, pancreatic amylase. ये amylases जो है वो कौन सा enzymes में आ जाता है? They are coming under hydrolases. So amylase is an example of hydrolase. Here, in the presence of water, the starch in the amylase is helping in breaking down the starch. By breaking down the bond between the polysaccharide, we have learned glycosidic bond. Between the amino acids, what we have learned? Peptide bond. Between the nucleic acid, what we have learned? Ester bond, isn't it? These all are bond by dehydration. By dehydration, you know, water is removed and the bond is formed, isn't it? So this bond can be broken by addition of water. If you add water, this bond can be easily broken. So the enzymes hydrolases helps in breaking of the bonds by addition of water, and they are involved in reaction, especially the digestion reactions, where complex are broken down into simple forms. Okay, so these are hydrolases. Another category of enzyme is your lyases. So lyases, when you see, again they are also helping in breaking complex substances into simpler forms, but without water. Other than water, if the bond is broken, then the enzyme which is playing their role is what? The lyases. Okay, so this lyases, like for example fructose 1,6 bisphosphate, respiration mein fructose 1,6 biphosphate is when you are seeing, that is a 6 carbon compound is broken down. How many carbon is there? Fructose 1,6 biphosphate mein 6 carbons are there. Okay, so this 6 carbon compound is broken down into 2 molecules of 3 carbon compound. What is the 3 carbon? Phosphoglyceraldehyde is a 3 carbon compound. Okay, so one means here when you are seeing 2 things are produced, 2 3 carbon compounds are for you. What is that? One is phosphoglyceraldehyde, another one is dihydroxyacetone phosphate. So one 6 carbon compound is broken down into 2 3 carbon compound. Okay, so one 3 carbon compound is what? Phosphoglyceraldehyde. Another 3 carbon compound is what? Dihydroxyacetone phosphate. So 6 is broken down into 3 plus 3. Here aldolase is the enzyme used. It is a category of lyases enzyme where without water the bond is broken and 6 carbon compound is converted into 2 3 carbon compounds. Okay, so that is again lyases, breaking of bond, they are taking place. Next is your isomerase. What do you mean by isomerase? Isomerase means in this only the means one form of compound is converted into an isomer form of other compound. Like for example isomers, you know, isn't it? Molecular formula will be similar, structure will be somewhat different, C, O, H and all arrangement will be different, but when you see the formula is totally the same, like glucose as well as here fructose 6 phosphate and glucose 6 phosphate. So this is fructose 6 phosphate, this is glucose 6 phosphate. Okay, both when you are seeing they are having the formula the same but the structure is different. So this is isomerase. So when you are seeing somewhat they will be resembling the same carbon, hydrogen, oxygen and all will be present, but the distribution is somewhat different. So this sort of isomerase helps in isomerization reaction, in which one form of compound is converted into another form of the same isomers. Okay, so for example fructose 6 phosphate and glucose 6 phosphate. Glucose 6 phosphate is converted into fructose 6-phosphate. And what is the enzyme involved there? We call it as hexoisomerase. Otherwise, we can also call it as what? Your glucose fructose isomerase can also be called hexose. Hexose 6 carbon is present. That's why we can also call it as hexose isomerase. Okay. So isomerase helps in what? Isomerization means the same, just conversion of the structure, but the function remains the same. The last form of enzyme is your ligases. Okay. Ligases, when you are seeing ligases here, joining is taking place. What is taking place? Joining. So first one is oxidoreduction, oxidant reduction, transferation, transfer of group, hydrolysis helping in breaking the bond by addition of hydrogen, helping in lysis, helping in breaking the bond without water. Okay, here with the water. Here isomers are just interchanging, and thereafter ligases, they are helping in bonding, means they are making the bond and forming bigger compound. Okay, joining is taking place. So for example, here it is given pyruvic acid or pyruvate. Okay, Pa is what? Simply we are writing pyruvic acid, pyruvate is combining with carbon dioxide. With what they are combining? They are combining with carbon dioxide. Photosynthesis and all will be learning. Okay, so this pyruvic acid, when they are combining with carbon dioxide, this pyruvic acid is a 3 carbon compound. Carbon dioxide is a 1 carbon compound. 1 3 carbon compound combines with 1 carbon compound to form oxaloacetic acid. What is OAA? It is oxaloacetic acid, which is a 4 carbon compound. So here one is combining with that of the other, forming bond, and joining is taking place, because of which a complex substance is formed. So here this one is called as what? Enzyme which is involved is pyruvate carboxylase. Carbon dioxide is added. So carboxylase is added to pyruvic acid to form a 4 carbon compound, oxaloacetic acid. Hope so it's clear. There's the 6 classes of enzyme. I think most probably you would have understood. In your book mostly it will be given like this. Oxidoreductase, 2 substrates are there, isn't it? So one is getting here it is in the reduced form, oxidized form. When the reaction is taking place, S is getting oxidized, isn't it? S is getting oxidized and S dash is getting reduced, just interchanging. One is oxidized, another one is reduced. Okay, transferases, here S is having the group. Okay, this S group, the functional group amino group is transferred to what? S dash. So what it will happen? It will convert it to S plus S dash G. Okay. So actually transferases, when you were learning, I will take out, where is that one? Yeah, transferases, when you are learning, you have learned this glutamic acid was having what? The amino group, it is transferred. So group यहाँ पे होता है, ये group यहाँ से कहाँ पे चला जाता है? Pyruvate पे चला जाता है. ठीक है. So automatically when you are seeing this amino group is transferred from one to another. So in transferases, the group is getting transferred from one to another. So like that the change will take place. Okay. Hydrolysis, you can find the breaking of bond is taking place. Hydrolysis, okay, catalyzing hydrolysis, breaking of bond is taking place with the help of water, and the break is taken place and has converted into small monomers or means 2 molecules like that. Lysis, again breaking is taking place, okay, but without water when it is happening, other than hydrolysis, without water when it is involved, then we call it as what? We call it as lyases. Okay. The next is your isomerases, interconversion, optical isomers, just the positional isomers changing from one compound to another. Ligases, what they are doing? They are helping in linking together of 2 compounds. 2 compounds are combining together. Okay. So this is what which is given in your NCERT, but with this example if you learn, it will be better. But for writing, if you are learning this one and writing, it's fine. But for understanding, I think this will be much helpful. Okay, so this one you can understand easily. So oxidoreduction में एक तो oxidize हो रहा है, ये reduce हो रहा है. Okay, transferases, amino group यहाँ से यहाँ पे चला जा रहा है. Hydrolysis, with the help of addition of water, starch is broken down into smaller ones. Lyases, without addition of water, fructose is broken down into 2-3 carbon compounds. Isomerases, just the position is changed. And then ligases, joining is taking place to form a bigger one. Okay, with this biomolecules is coming to an end. Hope so it's clear, and if at all there is any doubt, please feel free to ask me. Okay, so I will wind up for today. Thank you.